Okay, any questions?
I had a few questions about the lab assignment from Tuesday.
Okay. If it's really long, I may have to interrupt it and say we'll discuss this at some other time, but if you can certainly start.
I was just curious, like the best way to go about finding the answers for the case study portion. Like is a bit of a hint would be to like look at like the stain and try to figure out like what bacteria that is, like depending on whether it's gram positive or negative or something like that.
Well, the case studies are very esoteric, so you can use whatever you want to figure out the case studies, but first read the case study and see if that gives you a hint as to what it is. And then take a look at the data they did. The data they show gives you obviously what the answer will be. And look in the lab module, especially in the tables, for determining what the answer is. I don't know if that helped very much or not.
Yeah, it did. Thank you. I'll take a look at the tables and see if that helps me out.
Okay.
Thank you so much.
Obviously, the results that they give you is enough to get the answer, but you have to understand the results and how to interpret it, I guess.
Okay, gotcha. Thank
Okay.
you.
All right. Any other questions? I noticed there's a few emails. I have not looked at my email yet today, and I was hoping to do that in between classes, and sorry, I didn't get to it. If there's no further questions, let's begin. Can you see my screen?
No, we cannot see it yet.
Okay, thank you. Make sure I'm recording. Looks good. All right, today is July 21st, and we're supposed to be covering Lab 5, Streaking for Isolation and Culture Media. You should also read the unknown project instruction files on Canvas for next week, because next week I'm going to be discussing the unknown project. So gosh, we are already in week 3. Whoops, the lecture just finished in the Zoom program. So let me save this down. And obviously I'm not going to get it posted right away, but hopefully after the lab, I'll get it posted. You won't be able to see it right away, but I think a half an hour, an hour after I submit it to YouTube, you can then view it. All right, let me show you the unknown project instructions files. Oops, I think it's under week 5. Let me look to see if that's turned on. Nope, let me turn this on. We'll need that. Not for this week, but for next week. I think everything else is turned on. And we're not using week 9. All right. Oh, that's the journal. Here it is right here, the unknown project instructions. So please read this. I will discuss it, but it'll be good if you have some understanding before I begin discussing it. You do not need to read the sample journal, the building a dichotomous game plan, and the unknown notes, and your unknown report rubric, how the unknown project will be graded. But you should look at the unknown instructions, and I'll be discussing each of these, mainly this one, but we'll briefly look at that, and I'll look at the this and And this, you might want to take a look at your unknown list. This is the different species of bacteria that we will be working with when you're working on your unknown project, where you will be given a bacterial species, and then your goal is to use different tests to identify the species. So here on the left, we have the different species that it could be. And on the top, we have the different tests you can run. It runs further than this is showing. There we go. The different tests that it can be. And you have to identify the different species. And how we're going to do this is I'm going to give you a bacteria number, which is going to represent one of these bacteria species. And I will give you the first 3 tests, but after that, you have to pick a test that you want to run to try and determine what your bacteria is. Go back to, here is it? And you have to use a dichotomous game plan. We'll talk more about that next week. So you simply can't tell me. Where do I have it? You simply can't tell me, give me the results to all of these tests. Because I'll write back to you and say, we're supposed to be doing a dichotomous game plan, so you can't ask for the results to all of the tests. Okay. All right. Any questions about any of that? That's just an introduction. We will discuss the project next week. And I want you to read the instructions so you will be better able to do the project. And then you'll be able to understand what I'm talking about when I'm talking about the project. Any questions about that? All right. So let's move to lab 5, streaking for isolation and culture media. Note, we're already in the end of week 3. We're moving very rapidly here. We always do in the summer, where the class is truncated. Next week, we'll be halfway done with the class. Kind of scary when you think about it. Any questions about any of that? All right, let's go into the lab. Doing lab 5. Should have had these up to begin with. Oop. Remember, always put your answers on the worksheet and submit your answers in the worksheet. Do not submit the lab module. If you do submit the lab module, you'll get a deduction, because I don't want the lab module. All right, lab 5, cultivation of bacteria, culture media, inoculation, and isolation. You do have some pre-reading, it's not very much. And then there are some video clips. I don't think we have a virtual lab. Let me check at that. I think the virtual lab's crossed out. So I haven't found another virtual lab that will run. So there is no virtual lab today. The primary learning objectives: upon completion of the lab, students should be able to understand the importance of proper specimen collection and transport to the lab, be able to explain the 5 principal steps used by microbiologists to process specimens, differentiate between the various types of culture media. So we'll talk about different types of culture media, and you need to be able to differentiate between them. Be able to explain the purpose of streak plate culture technique, AKA streaking for colony isolation, and then be able to define the terms. What's the selective media, differential media, and reducing media? The ability to isolate pathogens from a clinical specimen submitted for the culture is only as good as the collection technique and the timely transport of the specimen to the lab. Some microbes are sensitive to desiccation, meaning they'll dry out, and others are exposure to oxygen, that's the anaerobes, and require a special transport medium to preserve the specimen. Once a specimen is received in the lab for microbiological culture, microbiologists employ the 5 principal I steps to process the specimen. First, they inoculate it, then they isolate it, they incubate it, they inspect it, and then they identify it, meaning identification. Inoculation is placing the specimen on an artificial culture medium to initiate its growth. They isolate it by spatially separating microbial growth into distinct colonies. You need distinct colonies so you'll have a pure culture. And usually you do that by sending the culture through 2 colony isolation events. You incubate it, and that's just placing the culture medium in an environment where the conditions are facilitated so that growth occurs. Normally, we incubate bacteria in an incubator. We then inspect the culture, examining it for the presence of microbial growth. You can also do some other characteristics, but you usually can't identify it just by looking at the culture. You then begin the identification, and here you perform specific tests to identify the pathogenic microbe that's been isolated. Okay, the 5 steps will be discussed, but first we have to discuss different types of culture media. You're already familiar with general purpose media, and that is to grow many bacteria cheaply, such as growing it on Na plate, nutrient agar, on nutrient agar with yeast extract or Na plates, or growing it on TSA, tryptic soy agar. These grow many different organisms, not all organisms, but many, and there's no selection on the plates. And their purpose is just to grow the bacteria and to grow them cheaply. They do not work, general purpose media, for fastidious microbes, organisms that have specific nutritional requirements. And you have to put something in the media before the bacteria could grow. We could put blood in the general purpose media, in which case we call it an enriched general purpose media, where we add 5% sheep's red blood cells to the media to help organisms grow. For example, Streptococcus pyogenes, which causes strep throat, will not grow on a general purpose media, but it will grow on a blood agar plate. And that's an enriched general purpose media that contains, I don't remember if it's TSA or Na, I think it's TSA, and then 5% sheep's blood. And then the red blood cells provide the nutrients that Streptococcus pyogenes requires in order to grow. Any questions about any of that? A selective media is one that contains chemicals that inhibit or halt the growth of some organisms, but allow other organisms to grow. Keep in mind two things when you're using a selective media. First, the selection is not necessarily perfect, meaning you might get reduced growth, but you do have some growth. And second, those organisms that are supposed to grow on the selective media may not grow as well on the selective media as they grow on another media. So usually when you're growing on a selective media, you compare the growth to the growth on a general purpose media. All right, any questions about selective media? No? Let's move on to the next media then, and that is a differential media. A differential media is a media that contains chemicals that produce visible differences between two different species. These differences may just be in the coloration on the plate, but they could be other differences that you can distinguish two different species, the way they look. The differences are unrelated to how well they grow on the differential media. But you can look at the plates and then distinguish differences between two different species by a differential media. I think I've got an example here in a bit. The fourth type of media is a reducing media. This is a media to promote the growth of anaerobic bacteria. Anaerobes either grow in low or no oxygen conditions. And in order to get an anaerobe to grow, you have to grow it in a reducing media. Now, the reducing media removes the oxygen. It's important to note that a culture media can be classified into more than one category. For example, MacConkey's agar is both selective and differential. Blood agar is both enriched and a differential medium. Any questions about any of that? Excuse me. More of a tickle than blowing my nose, but all right, let me show you MacConkey's media. Here we're looking at MacConkey's media. It is selective in that it has ingredients that select against gram positives, and the gram negatives can grow on the MacConkey's agar. You don't need to know what the ingredients are at this point, although some point in the future you might need to know. But MacConkey's agar has bile salts in it and crystal violet in it. And both of these prevent gram positives from growing. The crystal violet is a stain which actually stains gram positive cells, and the crystal violet gets in the cell wall and then prevents the cells from growing. Bile salts are something that are found in the intestine where we have a lot of gram negatives. And so gram negatives, well, some gram negatives are used to growing in bile salts. And so they will grow in MacConkey's agar, and many gram positives will not grow in the presence of bile salts. So MacConkey's is selective, and if it grows, then it's a gram negative. If it doesn't grow, or the growth is very poor, then it is a gram positive. And I guess I should point out that if it's selective, the growth may happen, it just will be poorer than if you were to grow it on a general purpose media plate. McConkey's, the selection is pretty good. I've never seen a gram-positive which grew well on McConkey's agar. There might be one, but I've never seen it. All the gram-positive I've seen generally don't grow on the McConkey's agar. So the selection is pretty good, but usually it's not that good for selected media. For McConkey's it is. And then if it grows on the McConkey's agar, you can then differentiate between a lactose fermenter, which makes the cells stain pink, and then one that is a non-lactose fermenter, meaning it cannot ferment the sugar lactose, and then the coloration will not be pink. Okay? This only works, the differential, if the bacteria can grow. If the bacteria is selected against, you cannot determine the lactose fermentation ability of the cells. Any questions about McConkey's agar? All right, here we're looking at blood agar. It is both a enriched media and a differential media. It's enriched because if the bacteria cannot grow on a general purpose media, it might grow on blood agar if the blood supplies the nutrients required for the organism to grow. Like Streptococcus pyogenes can grow on a blood agar plate, but it will not grow on general purpose. So that's an enriched. Blood agar is also differential because it allows you to differentiate between hemolytic activity. Here we have bacteria shown in white by the gamma, and this is showing gamma hemolysis, where the blood is red all the way from, I don't know, anywhere on the plate, all the way to where the bacteria are growing, meaning gamma hemolysis, which is also no hemolysis. Here we have alpha hemolysis. Let me blow that up a little. And you'll note the blood is red, but before we get to where the bacteria are, which are off white or white color in the alpha, we have a dark ring around the bacteria. The dark ring are from alpha hemolysis, which is also partial hemolysis. You will see a darkening of the blood with alpha hemolysis. And it's really specific. All of the inside of this is darker than right there, and that's from partial hemolysis. If it isn't darkening the blood, it may look greenish. So if the blood looks greenish around the bacteria, that is from partial hemolysis, either darkening or greenish. Okay, any questions about any of that? All right, the third hemolytic activity is complete hemolysis, which is called beta hemolysis. Here in white, we have the bacteria in the B, and then around the B, we have a complete clearing of the blood. So right there, it's not red, it's clear, because all the red blood cells have been lysed by the beta hemolytic bacteria shown in white. And this isn't white, that's clear. If you were to hold this plate up to your eye, you could actually look through it right here. And obviously wherever it's clear. You couldn't look through where the beta is because the bacteria will stop you're being able to see through it. And then right here where the blood is, you can't see through a blood agar plate. Any questions about any of that? So the three types of hemolysis, gamma, alpha, and beta hemolysis. Normally, when you have beta hemolysis, you'll have a darkened ring around the beta hemolysis. This right here is from partial hemolysis. And that's normal in a plate. Any question about any of that? One more time, I think. All right, this table shows you the general purpose media, the different types of general purpose media, and then some comments about the general purpose media. Both gram-positive and gram-negative bacteria grow if they're non-fastidious. An enriched media plate, and this can be also differential because we're only talking really about blood agar, which is both differential and enriched. Promotes the growth of organisms like Streptococcus pyogenes. It's differential on the basis of the hemolytic activity of the blood. CAP is an enriched general purpose media. It is not differential, so it's only enriched. CAP, chocolate agar plate, differs from BAP, blood agar plate, only in the temperature that the agar is when you add the blood. Meaning, if you add the blood when the agar is still hot from coming out of the autoclave, you will get chocolate agar, where the temperature of the agar will lyse the red blood cells. And that'll make the agar brownish in color. And that's why it's called chocolate agar, because the plate is brown in color. Chocolate agar does permit the growth of some species of bacteria, like Haemophilus species and Neisseria gonorrhea. It does not permit the growth of Streptococcus pyogenes, is that right? I might have that backwards. I think it does permit the growth of Streptococcus pyogenes, because the nutrients are supplied by the red blood cells. Anyways, the BAP, you're adding the, or blood agar, you're adding the agar, not the agar, you're adding the blood after the agar has cooled sufficiently so it no longer lyses the red blood, but the agar isn't cool enough so that it will solidify. You only have about 5-degree temperature range that you need to add the blood to the agar. And it's someplace between something like 50 degrees and 55 degrees. I think that's Celsius. You have to add the blood at that temperature. Below that, the agar will solidify, and then you can't add the blood. Any question about CAP, meaning chocolate agar and blood agar? All right, both of those are enriched media. Blood agar is also a differential media. MacConkey's, EMB, and HE, meaning hectone agar, EMB, eosine, methylene blue agar, sorry, and then MacConkey's or MAC, are both selective and differential media. All of them are selective, and they select against gram-positive, so only gram-negatives can grow on these plates. All three of them are also differential, and they're differential on the basis of lactose fermentation. They will be a different color if they ferment lactose than if they do not ferment lactose. McConkey's, I can remember, is pink if it ferments lactose, and it's not pink if it does not ferment lactose. With Hectone agar or HE agar, it's salmon colored, meaning an orangish pink color, if it ferments lactose, and it's not salmon colored if it does not ferment lactose. Any question about selective and differential media? Okay. PEA, phenyl ethyl alcohol, is a selective media, and it selects against gram-negative cells so that only the gram-positives can grow on the PEA. I should mention the selection of this plate is not 100% perfect, so occasionally gram-negatives will slowly grow on the plate. Is that right? Yeah. It has alcohol in it, and that gets in the gram-negative cell wall, and that selects against the gram-negative bacteria. Any questions about PEA? And then there's mannitol salt agar. It is both selective and differential. It is selective in that it has 7% sodium chloride in it. Why it's called salt, mannitol salt agar, 7% sodium chloride. And unless the bacteria can grow in 7% sodium chloride, it'll be selective against. If you don't know, most bacteria do not like an environment higher than 2% sodium chloride, and this has 7%. It is also differential in the ability to ferment the sugar mannitol. And if the bacteria can ferment the sugar mannitol, the bacteria or the region around them will turn yellow. And if the bacteria do not ferment the sugar mannitol, the bacteria will not be yellow and will be the same color as the bacteria normally are. And then the media around the bacteria won't be yellow. It'll be the normal color, which is kind of a pinkish purple color. Pinkish color would probably be the best word. Staphylococcus aureus can be grown on mannitol salt agar, and it will show a yellow pattern around the bacteria, and that'll allow you to differentiate Staphylococcus aureus, which is a human pathogen, from other staphylococcal species, which can grow on mannitol salt agar, but it will not turn yellow, at least most of them, staphylococcal species. Okay. What else should I talk about mannitol salt agar? There was one other thing, and I'm drawing a blank here. I don't remember. Oh, here it is. It's called mannitol salt agar because it has the sugar mannitol in it, and it has sodium chloride in it. So why it's called mannitol salt agar. So this name tells you exactly what chemicals are in it. All right, the 5 steps that a microbiologist uses once they get a specimen, a clinical specimen, to grow in the lab is to inoculate it, put it on the tissue plate, usually disperse it so that you'll get different colonies, and then isolate a colony, streak it out for colony isolation a second time, get a pure culture of it. So isolate it to get the pure culture. And now when you do a test on the culture, it's a pure culture. So you know the results are from the bacteria of interest. Like if you did the test on this bacteria, you won't know, did this bacteria make the test positive, or was it this bacteria or this growth? I'm not sure, that could be a mold. So when you have a mixed culture, you don't know which bacteria caused the test to be positive. So you need to get a pure culture. Louis Pasteur developed the concept of a pure culture and then worked out a way to get a pure culture. His method was very complex. You had to use limiting dilution, meaning you got 10 tubes and you diluted your bacteria. And if in that dilution you only had one tube that had growth in it and 9 other tubes did not have any growth in it, then you used the bacteria growing in that one tube and said it was probably a pure culture because very few organisms were derived to cause growth in the tube. And how we know it was few, because 9 of the tubes got the dilution but did not get any growth because no bacteria were put in the tube. And if ever you got more than 1 tube out of 10, you then would have to use a lower dilution, or I guess a higher dilution, so that you would only have 1 tube out of 10 having growth. So if ever 2 tubes out of 10 had growth, Louis Pasteur would not use it. Okay. And this was very difficult because he had to make different dilutions and then put each dilution in 10 different tubes and then look for the dilution where he only got 1 tube out of 10 showing growth. It wasn't very efficient for developing a pure culture. Robert Koch developed an easy and efficient way for getting a pure culture. And what he did was he simply got a plate, a solid agar plate or solid plate, and then streaked the bacteria over the plate. And he streaked the bacteria to get isolated colonies. And then he would take from one isolated colony on the plate and then streak it on a second plate, getting isolated colonies. If ever he got a colony that went through 2 colony isolation events, he would say, I probably have a pure culture. And his chance of getting a pure culture is greater than 95%. This was a very easy and quick way to get a pure culture. And that's simply colony isolation or streak plate method. Any questions about any of that? So when you're doing the colony isolation event, meaning a streak plate for isolated colonies, what you do is you get your bacteria on a loop and then streak it out in one sector. And then you sterilize the loop in a Bacto incinerator and then streak out another sector. And as you're streaking, you make sure that your this sector has some overlap with the first sector so that you pick up some cells from sector 1 so that some cells will then be carried to sector 2. Okay, this isn't quite showing it correctly. Let me see if I can show it a little better. I'm going to close that. I won't be able to draw it very well because I'm not a good artist, but I'll get the idea out. So we have a plate, which would be a round thing like this. And what you do for sector 1 is you pick up your bacteria on the loop. It's obviously a sterile loop. And then you streak out sector 1. Let me go and divide this plate into 4. So you streak out sector 1, going back and forth about 30 times on the plate. And I'm not going to be able to show you 30 times because I don't have that good of control, although I'm doing okay here. And this is sector 1. This is sector 2. You then take your loop and put it in a bacterial incinerator, killing off the bacteria. And then you streak sector 2. What you do with sector 2 is you come into sector 1 a little bit, and you only do it 3 times, coming into sector 1. And then you do sector 2, doing 20 to 30 streaks across sector 2. I'm not doing this too well, but that's okay. I'm trying to speed it up so don't take all the time. You get the idea? And then you do the same for sector 3. Let's do it down here. I'm doing Roman numerals because I can do that much easier than drawing a 3. So you take your loop, put it in the bacteria incinerator, kill off the bacteria, and then you come to sector 3, and then you go into sector 2, picking up some cells right here and here, and then you go into sector 2 3 times, and then you do your streak for sector 3. Any question about that? So if you've done it correctly, you only go into sector 1 to set up sector 2, where the bacteria are diluted. I started here, so they'll be most dense right here. I make that nice and thick for very dense. Oops, that's not what I meant to do. You get the picture. And then as you're streaking along, this will be less dense bacteria. And so you take where it's less dense to set up sector 2, and then you streak out sector 2. And then you streak sector 3, picking up where the bacteria are less dense in sector 2 to start sector 3. And then you streak it out on sector 3, and it'll be most dense right here and least dense right here. And then to set sector 4, you come in to sector 3 right here about 3 times. And then you start streaking sector 4 here. Any question about any of that? It's important that you do not cross over between 4 and 1, because your bacteria will be most dense right here, least dense right there. Any questions about colony streaking? I'll leave this in case I want to talk about it later. The purpose for doing colony streaking is someplace along the line. When you're streaking, you will put down isolated cells. In this plate, we're getting isolated cells in sector 4, but it doesn't matter where you get the isolated cells. They could be in sector 4, sector 3, sector 2, or even sector 1. The point is you're getting isolated cells. And right here, we put down one cell or a few cells, which then grew to form a colony. And you take from this colony to streak out another plate. And then when you have isolated colonies on this second plate, you take from that isolated colony and you say that that is probably a pure culture because it's greater than 95% chance that it is a pure culture. If you were to only take your cells from one colony isolation event, there's only a 75% chance that this colony is pure, meaning this colony here and that colony there may have been formed from more than one cells, in which case it would not be a pure culture. And you have to have from an isolated colony, like right here, it looks like there are 3 colonies growing together. None of those are isolated. All right, that's the purpose of colony isolation, to get isolated colonies. And then you can pick from an isolated colony after you've done it 2 times to set up a pure culture. Even then, you're not guaranteed of a pure culture, but you have a greater than 95% chance. All right, so you isolated the bacteria species, and now we have a pure culture of it. You have to incubate that culture, and usually that means put it into an incubator, such as this, and it provides an environment for getting the bacteria to grow. In the lab, we have very simple incubators. All they do is they provide a temperature for the bacteria to grow, and then some humidity. Now this one is more complex, and it's probably providing specific air for the bacteria to grow, like maybe 95%, not 95, 5% carbon dioxide is supplied. You can provide specific oxygen amounts in the incubator, and there's other things you can control. You then inspect the bacteria and look at obvious colony differences. You can look at the colonial characteristics like the size, the shape, the margin, the elevation of the culture, and you can learn some things from this, but usually you cannot identify it. Okay? Like if you were to get large colonies that are yellow in appearance, growing, well, I shouldn't say growing on anything. It might be Staphylococcus aureus, but you'd have to do 1 or 2 more tests to prove that it's Staphylococcus aureus, such as get it to grow on the mannitol salt agar plate. And then you do the test to identify the species. And there's many biochemical tests you can do to determine the species. I showed you when we looked at the list here, all the different tests you can run. And it's something like 20 different tests. I didn't count the number of tests, but we have like close to 40 species of bacteria and all those tests. It's more than 10, about 20 different tests. There's more tests you can do, but we're not going to do them in our microbiology class. And then you look at a table to determine what your species is, depending on the table results. Let me briefly go through the table here. Of course, I won't be able to read it. Let me blow it up. That's not really helping. Maybe I can read it. Staphylococcus aureus, which is someplace in here, will grow on mannitol salt agar. Of course, all Staphylococcus aureus will. It'll be turning yellow, and you can see beta hemolysis. So it'll be yellow in color, so it'll be pigmented. It'll grow on mannitol salt agar, which I don't think is shown on this table. Maybe it is. And then it'll be beta hemolysis. Oh, there it is there, probably. And that's the only one it could be of these results. Any question about any of that? All right, let's move on. So know the terms. And then go through the lab. There's a video to watch on inoculation of an agar plate using streak plate culture technique. Only watch to about 4 minutes and 15 seconds, because those are the only parts of the video you need to watch. Note that in this video, they're using 3 sectors instead of 4. That's not a terribly important. Then watch little videos on beta hemolysis, alpha hemolysis, and gamma hemolysis. We have a video on MacConkey's agar. You only need to watch to 3 minutes and 50 seconds into the video. And then a video on mannitol salt agar. Okay? Any questions about any of that? Let me go to the worksheet now. And then answer the questions on the worksheet. Describe a natural circumstance where staphylococcal species of bacteria might benefit from salt tolerance. So if a staphylococcal species was growing and it's in an environment with salt, give me a natural circumstance where staphylococcal species will benefit from the salt. So you can't tell me when it's found on a mannitol salt agar plate, because that's not a natural circumstance. That's an artificial circumstance where we're growing the bacteria in the lab. So give me a natural circumstance. And 2, how might hemolysis benefit a hemolytic organism? Meaning, why does a hemolytic organism hemolyze the blood? What does it benefit from doing that? So tell me what the benefit is. Okay. 3, imagine you have a culture containing a single bacteria species. It may be one of several possible species. Describe a scenario where you use mannitol salt agar, blood agar, and EMB plates to identify the unknown bacterial species. That is, how would you use each of these 3 plates to help you identify the species? And I'll give you a hint. Each of these, mannitol salt agar, blood agar, and EMB, have 2 properties which can be characterized to help you identify the species of bacteria. So you need to tell me what they are for each of the plates. 4, what is the main goal for streaking for colony isolation? Tell me what that is. 5, what happens to the growth on a plate if you forget to sterilize the loop in between sectors when streaking for isolation? It's a good thing I kept this here. So we sterilize the loop at the beginning, grab our bacteria, and then streak out sector 1. But now we forgot to sterilize the loop, and then we start streaking out sector 2. And then the same thing for sector 3 and for sector 4. So don't tell me the bacteria will become contaminated because you forgot to streak in between the sectors. Between sector 1 and 2, between sector 2 and 3, you did not forget to streak or sterilize the loop before the sectors. So the plate will not be contaminated. Tell me what you will get. You need to tell me how the plate will look in sector 1, 2, 3, and 4. So literally tell me what you'll see in sector 1, what you'll see in sector 2, what you'll see in sector 3 and 4. So be specific, discussing each sector. 6, what happens to the growth on the plate if you transfer from the stock culture to inoculate each of the sectors? So here, sector 1 is always obtained from coming into the stock culture. Let me see if I can draw that. So this is our stock. Okay, well, what will happen to the plate if to set up sector 2, we grab cells from the stock and then streak out sector 2? And then what will happen when we grab from the stock and streak out sector 3? Okay. What I'll be looking for is a difference between 5 and 6. And if you discuss each sector, I should be able to see that difference. 7, suppose you successfully streak a plate to obtain isolated colonies from a mixture of 2 different bacteria species. How can you use this plate to create a new pure culture of only 1 bacteria? Be specific in your answer. 8, in the table below are a variety of agar plate media which you run on the bacteria. To note whether each media is non-selective, selective, or differential by placing an X in the column. You may use the table of media in the lab module to answer these questions. Okay, so nutrient agar, if you think it's non-selective, put an X here. If you think it's selective, put an X here. If you think it's differential, put an X here. And if you think it's all 3, put an X in all 3. And that's how you answer this question. 9, we have 3 plates here, and it's asking which agar plate was inoculated using a streak plate technique, meaning where you streaked for colony isolation. And then that's it. When you're done, type up your answers and submit it to the appropriate place for submitting worksheet for lab 5. And make sure you have it submitted by Saturday, 11:59 p.m. because I'll begin grading about that time. Any questions? If not, it's about 8 o'clock. I'll be here until about 9 o'clock to answer questions. But realize that when the last student logs out, I'm going to log out also. So far, students have always logged out early. Although last time, the last student didn't log out until about 5 minutes or 10 minutes before 9. But when the last student logs out, I'm gonna log off as well. All right, any questions? If not, go ahead and get started.